So my name is Tim Roberts, and I'm the site PI of the Simons Foundation 16p11.2 VIP project at the Children's Hospital of Philadelphia. And I want to discuss some of the advanced imaging findings that we have made in the 16p11 study, and relate those to some other findings that have been made in the broader field of autism spectrum disorders. I have some disclosures and funding sources for my research. And now I want to introduce the topic of the conversation. So in addition to behavioral and clinical assessments shown on the left of the slide, we also have insights into the brain of individuals with the 16p11.2 copy number variation that are available to us from various forms of structural, quantitative, and functional imaging. And so the topic of this webinar is to discuss these items. In order to do this, we want to introduce two technologies, and there's two machines pictured on this slide. That is the MRI, or magnetic resonance imaging device at the top, and also a technology called MEG, or magnetoencephalography, in the lower photograph. And with the combination of MRI and MEG, we're able to make images of the brain structure, also images of connections within the brain, and indeed the dynamic signals that represent brain activity. To go into a little bit more detail about this, we're able to look at, on the left, not just a static picture of the brain, in this case showing a cartoon of the auditory pathway drawn onto the MRI in the background, but we're also able to make images of the connections within the brain, the white matter wiring of the brain, suitable for putting on the covers of textbooks or such like. Some of the radiology or structural imaging findings are dramatic and readily visible, as seen on these images here, and as highlighted by the arrows. One might have an extension of the lower part of the brain called the cerebellum, or a cyst in the spinal cord, as pointed to by the red arrow. Similarly, white matter structures of the brain, this white structure in the middle of the brain is the corpus callosum. That's the bundle of fibers that connects the hemispheres of the brain, that normally has a certain appearance, can be sometimes a little thicker, particularly in the 16p11.2 deletion carriers, and sometimes a little thinner or even absent in the duplication carriers. So here is the first example of opposing effects, where we see something thicker in the deletions and thinner in the duplicates. And indeed, sometimes the effects are as qualitatively visible as this, or in the next example, one can see these black structures in the center of the brain. That's the ventricles, the CSF spaces of the brain, could be enlarged, in this case in the duplication. So sometimes it's as easy as this. But in fact, if you look at the population statistics, these qualitative observations are perhaps visible about a quarter of the time. So for the rest of the time, we have to go on a little bit more detailed investigation. So with MRI, we can go beyond simply looking at qualitative structural differences and look at measuring the actual volume of brain tissue. And so in this work from Randy Buckner, using computer algorithms to trace out the gray and white matter, it was possible to elucidate that in the group of individuals with a 16p deletion, we saw an overall increased intracranial volume, that is a slightly larger brain in the children with the deletion, and in fact a commensurately smaller brain in the adults with the duplication. So this is really just giving us some insight into the level of quantitative, not qualitative detail that can be obtained using three-dimensional MRI. It goes beyond just the whole brain. One can look at individual regions of the brain, in this case such as the hippocampus or the cerebellum or the thalamus, different regions of the brain that have different functions. And one can see, again, looking at the thalamus in particular, that the overall trends that were seen for slightly enlarged in deletions and slightly smaller in duplications, seems to hold out not just for the whole brain, but also for individual structures. So we're talking about maybe as much as 5 or 10% change in the anatomic size of these regions. However, size isn't everything. We're also interested in looking at the connections, the wiring of the brain. And this involves not just taking, let's say, a regular MRI, but a more advanced form of MRI called diffusion-weighted imaging. And diffusion-weighted imaging essentially allows us to image the white matter fibers. Fibers like celery, it's fibrous, so is the white matter of the brain, having a preferred direction. And in this study from Jeff Berman, it was possible to trace out both the early fibers of the auditory pathway from the thalamus to the auditory cortex in yellow, and also to look at the fibers of the language network in red. This is the arcuate fasciculus. And we can not only visualize these structures, but also interrogate the quality of the white matter bundles in terms of the orientation and degree of fibrousness and the ability to conduct signals. And again, in this example, Dr. Berman was able to show an atypical maturation of these white matter fibers in the children with 16p11.2 deletion, suggesting that the white matter was not sufficiently developed as age-matched controls, and therefore this might lead to downstream consequences in terms of signals propagating from one area to another, and that indeed might be underlying behavioral difficulties. Switching from MRI to MEG, we're able to look at not just the where in the brain question, but also when are things happening in the brain. Brain activity is a dynamic process happening in fractions of a second, and we're able to visualize with the MEG technology the spatial and temporal nature of brain activity, in this case in response to hearing a sound. The interesting thing that happens is after hearing a sound, about 1/10 of a second later, the neurons of the auditory cortex have a characteristic burst of activity, which we label the M100. And what we have previously observed in children with autism spectrum disorder is that this response is delayed by a fraction of a second, in fact just about 100th of a second. Nonetheless, these fractional delays in signals propagating through the brain can cascade and lead to a difficulty in understanding language. So whilst we have this finding of a delayed M100 in autism spectrum disorders, where did this come from? How does this relate to the MRI? And by combining that with the diffusion imaging, the fibrousness imaging that we showed before, we were able to suggest that, in fact, the quality of the white matter connections indeed did lead to the timing of the cortical response, even in a fraction of a second. And that led to our hypothesis that the diffusion imaging really is telling us something about the speed at which the white matter is able to conduct signals, so-called conduction velocity, and that as the white matter matures and the conduction velocity increases, we get a shorter time of arrival or latency of a response. That is part of normal development that seems to be delayed in children with autism. Similar responses, again revealed by MEG, have been able to predict, delays in these responses have been able to predict clinically relevant aspects of, for example, language impairment, that is performance on clinical language tests, suggesting that these timing parameters are truly significant, and that the longer it takes for the brain to respond, the weaker the performance is on a language test. And that's what's shown by this downward slope here, is that timing of neural signal communications really predicts clinical language score. And this was all in children with autism, but what we found in the 16p11.2 population is that the children with deletions in the two yellowy green bars in the bar chart on the right showed delays compared to controls and to children with the duplications, very similar to what we see in the autism spectrum disorder. That's not to say that these individuals met diagnostic criteria for autism, but rather that they share a mechanism whereby presumably atypically developing white matter, as shown on the diffusion imaging, led to a delay in the timing of the neural responses to the neural communication, as though perhaps there was traffic on the highway or construction on the highway leading to a delayed transit time on a journey. And even though this is only in fractions of a second, it's quite possible that this is underlying the phenotype of difficulty with processing spoken language. So in this case, we're seeing that the children with 16p11.2 deletions have a delay in the neural response, very, very similar to what we have observed in children with autism, and perhaps attributable to the same underlying white matter difficulties in development. An interesting aspect of this is that as children get older, typically their M100 response, that's this neural response, indeed matures and gets faster and faster, and that's evidenced by a downward slope on these plots here. The blue lines represent typical developing children, and the red, the children with the 16p11.2 deletions, green, the duplications. And we notice that, in fact, in all three groups, there is a downward slope, meaning that there is, in fact, a maturing process underway, and that these responses are indeed getting faster and faster with development, which is encouraging. Similarly, we are associating in autism the delays in neural communication as being consequent or secondary to the white matter maturation. And here we're showing plots that suggest that the similar thing must be true in 16p copy number variations, that as the white matter matures and the properties of the fibrousness of the white matter matures, we see a commensurate speeding up of the neural responses, and that's evidenced here by the positive slopes in all three colors. Again, suggesting that the same rules probably do apply in 16p11, that indeed maturing white matter is the basis for maturing neural communication and speeding up of responses, and that the delays in responses that we see are improving with age and improving with white matter maturation, although still a fraction of a second delayed compared to typically developing controls. So in summary, what we have here is a story that links genetics through the brain to behavior, and we have borrowed a lot from our understanding of autism spectrum disorders, and we have certainly had to combine both structural and connectional imaging with white matter quality and development and maturation with functional findings like the timing of neural signals. What we see with both the structural and the functional imaging is really quite commonly effects that are different from typical development in both 16p11.2 deletion and duplication carriers. And often, but not always, these effects are going in opposite directions, suggesting that there's perhaps an effect of what we call gene dosage. Certainly what is clear that it's necessary to take multimodal approaches, that is, looking at structure, connection, and function in order to increase our biological understanding. And perhaps of importance, it's worth noting that our understanding that we're learning from investigation of the 16p11.2 population may have generalizable mechanistic interpretations that extend beyond this particular CNV to a broader field of neuropsychiatry. So I'd like to very much thank the participating families and my large array of collaborators on this investigation. As we have taken a very multidisciplinary approach, it certainly takes a village. Thank you for your attention.